Talking about protein synthesis goes with 14.1 through 14.3, and protein synthesis is how we go from a gene on DNA and turn it into a protein. So this part right here is protein synthesis, and in general it's made up of two larger parts: transcription and translation. In transcription we go from DNA to mRNA, and in translation we go from mRNA to protein. So today in this video we're going to look at transcription part first, DNA to mRNA message. So after our discussion last time about DNA replication, scientists began to wonder how DNA disseminated its instructions. So all the instructions that the cell needs to do, all the things it needs to do, is in DNA. But what exactly do those instructions tell the cell to do? So these two scientists, named Beadle and Tatum, won the Nobel Prize in 1956 for what they called the one gene, one enzyme hypothesis. They thought each gene on a chromosome encoded for a particular enzyme. So they thought it took a specific gene to make an enzyme that catalyzed one chemical reaction in their body, second gene for a second enzyme. And this was okay, but later scientists discovered that many proteins weren't enzymes. So they changed it. Instead of one gene, one enzyme, they changed it to maybe one gene, one protein. So in this explanation, one gene, one protein, every gene instead of an enzyme is going to code a different protein. Some of them are enzymes, some of them aren't. This too is inaccurate or incorrect, because many proteins are made up of several polypeptide chains that each gene can, and each gene can make several different polypeptides depend on where we splice them. So even one gene, one polypeptide isn't completely correct. The idea that one gene, one protein hypothesis is okay for the most part, but it's not 100% correct. We do want to remember, though, that genes, however, do code for the creation of proteins. So gene to protein. Not a specific protein, so it's not one gene, one enzyme, one polypeptide, not even one protein, but one protein precursor, we could say. And in our case, we are going to deal with the gene to protein route. So, protein synthesis in an overview. So, protein synthesis describes the process of turning genetic instructions, encoding DNA sequences, into polypeptide chains. So, we're going to look at the one gene, one polypeptide hypothesis in this class. There are four smaller parts. I talked about transcription and translation. So, those are each parts. But after transcription, there's something called post-transcriptional processing, and after translation, something called post-translational processing. That's when we just fix up or modify the products of transcription and translation so that they can work correctly. So first, DNA is transcribed into mRNA, which is a messenger. DNA is both too big to fit through the nuclear pores in the nucleus and get out to the ribosomes in the cytoplasm, and it's too important. We can't go sending DNA out to ribosomes to tell them what proteins to make, because if the DNA gets destroyed, that's it for the cell. So the cell makes mRNA, or messenger RNA, to pass the message, genetic message, the codon from DNA to ribosomes in the nucleus, in the cytoplasm. mRNA, second, is processed for its journey to the ribosome. Third, the message carried by the mRNA sequence is translated into amino acid sequences into proteins. And fourth, the amino acid sequence is modified to make a protein. So this is basically protein synth right here. Start out with DNA that it serves as a template that through transcription makes our mRNA message. Remember, it's RNA, so we have U's instead of T's. U pairs with A instead of T. There's no T in RNA. And then in translation, we take this mRNA and translate it into an amino acid sequence, a protein. So first, we can view it also as, this is bacteria, in eukaryotes, transcription of DNA to pre-mRNA, and then RNA processing into full mRNA happens in the nucleus. mRNA goes through the nucleus to the cytoplasm, to the ribosomes in the cytoplasm, where translation happens. This process right here is called the central dogma of biology. DNA to mRNA to protein, central dogma of biology. And that's important to remember. So please remember, central dogma of biology, DNA to mRNA to protein. Alright, so the first part about transcription is initiation. We're going to get started with transcription. So, transcription is the creation of mRNA. Transcription is the creation of mRNA from DNA. RNA polymerase. So we're going to make RNA just like we made DNA and DNA replication. We're going to use DNA as a template and build an mRNA chain off of it. And to do that, we're going to need an enzyme named RNA polymerase. It creates the mRNA chain, one nucleotide at a time, just like DNA polymerase creates a new DNA strand, one nucleotide at a time. So RNA polymerase does basically the same thing that DNA polymerase does, except for RNA. RNA polymerase we're going to start this transcription. RNA polymerase starts transcription at a promoter region. So here, there's a region on the DNA called the promoter. This is basically the start signal. It tells RNA polymerase where to start transcribing protein, or to transcribing mRNA. And that RNA polymerase attaches to this special part of the promoter called the TATA box. Why it's called that is there's the TATA sequence repeated over and over and over. That's what tells the RNA polymerase to bind here and to start transcribing mRNA. Other proteins called transcription factors bind onto the promoter region that help the mRNA polymerase onto the DNA and to start working. So our RNA polymerase recognizes the TATA box on the promoter or start region and binds onto it with the help of transcription factors. That's initiation of transcription. Now we're ready to start. Alright, in elongation, RNA polymerase synthesizes a long mRNA chain by adding RNA nucleotides one at a time that are complementary to the DNA bases. Just like in DNA replication, where DNA polymerase is making a DNA chain, new DNA chain based on the DNA template strand, RNA polymerase is going to do the same thing. So RNA polymerase, once it's attached to the promoter region, is going to start moving down the DNA chain in a 5 to 3' direction. Remember this. This is just like in DNA replication. Moves in a 5 to 3' direction. synthesizes in a 5' to the 3' direction. That means the 5' of the new mRNA, and it's adding on to the new 3' end. So, unlike DNA replication, RNA polymerase can unzip the DNA all by itself. It doesn't need helicase to do that. So, it's going to attach to the promoter and then start going in the 3' direction and add one nucleotide at a time onto the growing chain that matches up with the parental DNA. So the C RNA is going to match the G DNA, the A RNA is going to match the T DNA, but this time, if there is an A DNA nucleotide, we're going to put U or uracil in there instead because there's no thymine in RNA. Only one strand of DNA is used to create RNA via an RNA polymerase. So it's not making two mRNA transcripts, one on the top and one on the bottom like there's leading and lagging strands. No, there's only a leading strand for RNA synthesis. That means only one of the strands of DNA is used to actually create the mRNA copy. And that strand of DNA is called the coding strand. So this strand right here, that'd be called the coding strand. Perfect. Lastly, we're going to terminate, or we're going to stop the transcription. In termination, transcription stops, and the mRNA floats free from the RNA polymerase, and RNA polymerase detaches from the DNA double helix. In eukaryote cells like us, RNA polymerase runs into what's called a polyadenylation signal. It's basically a AAU, AAA, AAU, AAA, or in DNA it would be TTA, TTTTT, TTA, TTTTT signal on the DNA. Downstream from that, the RNA polymerase detaches from the DNA and mRNA is cut free. So, as soon as RNA polymerase runs across this polyadenylation signal, it's signaling it to stop and to detach, and we're done creating our transcript. The coding region that we're going to transcribe into mRNA, it's stopping, we're all done. After that, we need to modify our mRNA. So post-transcriptional processing is like mRNA modification. We need to get this mRNA ready for its journey to the ribosomes. We don't want it to degrade or get torn apart before that. So first, a modified guanine nucleotide called a G5' cap is added to the 5' end of DNA. So to the 5' end, we add a guanine triphosphate, or GTP molecule. It's a guanine with 3 phosphates attached to it. We add it to the 5' end. It's like a cap. It's sealing in the DNA. And on the 3' end, we add a poly A tail. It just is basically a bunch of adenine nucleotides repeating. And we add these to the 3 and the 5' end for 3 reasons. They protect the DNA, or the RNA, sorry. They protect the RNA on their journey. They aid in ribosomes recognizing and binding to this RNA once it's at the ribosomes. And it helps expedite the transport of this mRNA out of the cell. Okay? Lastly, RNA needs to be spliced. So I don't know if you know, but large sections of DNA don't code for any proteins at all. And those sections of DNA are still in the mRNA after we transcribed it. So what we need to do is cut those out. And these regions that aren't expressed, don't code for proteins, are called introns, because they're in between coding sections of DNA. So splicing is done by this complex called a spliceosome with the help of catalytic RNAs called ribozymes, but basically the spliceosome recognizes these introns, these non-coding parts, and cuts them out and splices what's left, the coding parts of DNA called exons, because they're expressed, back together into a finished product of post-transcriptional processed DNA. So our spliceosome cuts out these introns that we don't need, that don't code for any proteins, and splice together the exon together that do code for proteins, and then off it goes to the ribosome. All right, this is transcription. We're going to look at this in class on Friday. Have any questions, just let me know.